So for today, we're going to talk about experiment number 6, okay, which is called polymerase chain reaction and gel electrophoresis. So polymerase chain reaction, or as abbreviated PCR. So PCR, okay, it stands for polymerase chain reaction. These are the objectives for today. So the first objective is to define what is PCR, polymerase chain reaction, and determine its components. We also want to know its components that we put in the reaction. Second, list factors that affect PCR. Third, describe visualization of DNA by using a technique called gel electrophoresis. Yes. So, as I said, this experiment is a continuation of last week's experiment. Let's say we got the DNA from the patients. We want to determine now, we got the purified DNA, we want to determine which patient or patients they have the CML type of leukemia. Okay, the CML type of leukemia. So we got the blood sample. We isolated DNA from blood using that kit we talked about. We have pure DNA from each patient. Now we're going to be using a technique called PCR. PCR to amplify, to make photocopies of a specific target gene. Okay, we want to determine which patient has this gene, which is CBR-ABL, cancer gene that is present in leukemia samples in CML type of leukemia. Okay, this gene has a specific size. So we're going to amplify it. We're going to make many copies of it if it is present. If it's not present, it will not show up. Okay, so we're going to photocopy this gene using PCR, using a technique called PCR. And then, so we make many photocopies, but we have to use gel electrophoresis to visualize the DNA. Okay, so there's a technique called gel electrophoresis that enables us to visualize the DNA under UV light. So what is PCR? What is polymerase chain reaction? Now, PCR, guys, it's a technique, molecular biology techniques that has revolutionized the field of molecular biology. It's really a powerful technique that has changed the field because it has many uses, many different uses in many different fields. So these are just what you guys see here are just some examples. So it can be used to clone a desired gene. Let's say we want a desired gene, an eye gene or a hair color gene, etc. So we can amplify this gene, we can make many copies, and we can clone it to study it, to analyze it. If we want to study a cancer gene, okay, we can amplify it, we can photocopy it, okay, and then we can look at it, we can investigate it. So it basically allows us to photocopy a specific disease gene, a specific cancer gene, so we can look at it in more details. Okay, it can also enables us to amplify all DNAs, for example, from patients who have passed away a long time, but we have tissues from them stored in the freezer, let's say, so we can get this DNA and we can examine their DNA. We can use it to detect bacterial or viral infections, diagnosis, and in forensics and paternity testing. So it encompasses different areas. It encompasses different areas, different uses. It's a very powerful technique, extremely powerful technique. Now, PCR, or polymerase chain reaction, it was developed in the 1980s, specifically in 1985 by an American biochemist. His name is Kary Mullis. And for this invention, because it changed the whole field of biology, molecular biology, he actually won the Nobel Prize in 1993. So it's a very powerful technique that revolutionized the whole field. And it's a technique for in vitro amplification of specific DNA sequences. So here in vitro means in the lab. In vivo means inside human beings or inside the cell or inside the organism. So in vitro means in the lab. So we can amplify a specific DNA sequence like a gene in the lab. And it's a reaction that amplifies a particular piece of DNA, okay, or a gene, as I mentioned before. And it's very powerful because it makes billions of copies of a specific target sequence, specific gene, in a matter of few hours in the lab. So in a matter of few hours, like 1 hour and a half, you can get billions of copies of DNA, of a specific DNA. How does PCR work? PCR has 3 steps, okay? It has 3 steps. Okay, these 3 steps are in one cycle. Here it says heating, cooling, and replication. This is oversimplified. the steps are actually denaturation, primer annealing, and extension. Okay, denaturation, primer annealing, and extension. Okay, and this is repeated several times. Okay, so we have, let's say, 36 cycles. So one cycle is repeated about 36 times, 37 times, and this gives us this takes about 1 hour and a half and gives us gives us billions of copies of a specific pieces of DNA. Okay, so let's say we are looking for that cancer gene. We take the DNA that we purified from last class, and we basically amplify that cancer gene specifically, okay, after 1 hour and a half by going through these different cycles. What are the components of PCR? So let's say we have the purified DNA from last class, which is the DNA from the patient. The DNA from the patient, okay, from the blood that we obtained last class, we got the DNA. It's purified DNA. We want to see if the patient has the leukemia gene or has the specific gene. Now, this sample that we got from the patient, guys, we have the 46 chromosomes, right? The 46 chromosomes, there are many genes on each chromosome, but we only want that leukemia gene. We only want that CML leukemia gene. And this is what PCR does, okay? So to do that, we need to add the different PCR components. So the first component here is the patient sample. This is the double-stranded DNA that we obtained from last class. We call it the template. Okay. We also need to get primers. Okay. I'll talk more about primers, but primers are basically small DNA sequences that we buy. Okay. We buy from companies that specify the beginning and the ends of where we should amplify the DNA. So they are specific to the cancer gene. Okay. We make them in the lab, then we add them so they can only amplify the specific gene. So they are important for specificity. And then we have an enzyme, okay, a DNA polymerase, a special DNA polymerase that is thermally stable. Okay, thermally stable. We'll mention later on. It's actually called the Taq polymerase, which is important for the extension part of the cycle. Then we have the different nucleotides. We have the parts. Okay, deoxy, D stands for deoxy, different nucleotides, ATP, GTP, all the different nucleotides. Okay, and then we have the reaction buffer. So the reaction buffer, it's basically food for the enzyme. Food for the enzyme. Okay, the enzyme here, Taq polymerase, needs food, so it has salts, etc. Okay, so consider, guys, that we want to build a new house. Okay, we want to build a new house. Okay, this is the template for building a house, the location for building the house. The primers, they specify where we're going to build the house. The DNTPs are the building blocks, the bricks for building the house. That Taq polymerase, this is the worker who's building the house, okay, who's going to carry the bricks, who's going to carry the blocks, which are the DNTPs. And this worker cannot survive. When he builds a house, he cannot survive without food. Okay, and this is the food. Reaction buffer contains the right pH, contains the salts that are critical for the Taq polymerase for the enzyme to function. What are primers? Primers are basically oligonucleotides. Okay, there are small DNA sequences, about 18 to 24 base pairing, and they are single-stranded. Okay, and they bind specifically to a specific region. They bind specifically to a specific region of DNA by base pairing. Okay? So they act as the starting point, the beginning and the end. Okay, the beginning of the cancer gene and the end of the cancer gene, for example. Now let's talk about this cool new enzyme. Okay, cool new enzyme that functions at high temperatures. It's a thermostable polymerase. Okay, as you guys know, most enzymes, they function at 37 degrees. This enzyme is actually stable. It's heat stable. Okay, so it's able to survive, okay, at high temperatures because, as you know, PCR has several steps, including denaturation, and denaturation occurs at 95 degrees. So this should kill an enzyme, but it doesn't in our case because we use a heat-stable enzyme. Okay, it's taken from thermophiles. Okay, thermophiles. Okay, these are organisms that are able to survive at high temperatures. And here, specifically, the name of this enzyme is Taq polymerase. It's the type of DNA polymerase that is heat stable. It's called Taq polymerase. And its optimum temperature is 72 degrees, 72 degrees Celsius. What are these building blocks? These building blocks or bricks that the enzyme needs to build the new DNA, the billions of copies of the new specific DNA, they are called dNTP. Okay, dNTPs, and we also need a reaction buffer, the food for the enzyme. So DNTP stands for deoxynucleoside triphosphates. These are the bricks, and they're added by the Taq polymerase to the DNA, to the growing DNA. Okay. Now, the reaction buffer, it provides optimum condition for the enzyme, for the Taq polymerase, such as, the salt concentration of magnesium chloride to aid in the attaching of the primer to the DNA. Now, when we mix all of these components together, the Taq polymerase, the template, the dNTPs, etc., we put them in the PCR machine. We are ready to put them in the PCR machine, and then we set the cycles, and we'll talk more about the cycles later on. So we put them in the PCR machine, and we set the program here. So we tell it we need to go for 36 cycles, etc., of repeating denaturation, annealing, and extension. Okay, leave it for about 1 hour and a half, and it will give you billions of copies of the specific DNA. And then you can take the sample, and then you can proceed with gel electrophoresis. Okay. So what are these 3 steps of PCR? How do they work? Now, the first step is denaturation. Denaturation is where we take the DNA. So let's say, first of all, that we added all the components. Okay, so we took an Eppendorf tube, a microtube, and we added the template DNA from the patient from last class. We want to see if this patient has leukemia. We added the dNTPs, we added the Taq polymerase, etc. We are ready to go. So we put them in the machine and we turn it on. On the machine, you will see these different steps. So step number 1 is denaturation. And what happens in denaturation, it occurs at 95 degrees Celsius. The DNA template from the patient is denatured. So the hydrogen bonds between the 2 complementary DNA strands, they break. Okay, they break. So let's say this is one DNA strand and this is the other DNA strand. So because we have high heat, 95 degrees, the hydrogen bonds between the complementary base pairing, it gets destroyed, gets torn apart. Now, the second step is called primer annealing. Primer annealing, and this occurs between 40 Celsius to 65 Celsius. And in these steps, what happens is that the primers which we bought from a company, okay, let's say we know the sequence of the cancer gene beforehand, so we add these primers, which are single-stranded. We add 2 primers, primer 1 and primer 2, so that we know the end. We know beforehand the end of the cancer gene, the beginning and the end. So now we can extend this way. We can build the cancer gene. Okay, so they bind now to the single-stranded template DNA, and now we are ready for extension. Okay, primer annealing, guys, is very specific. This is the most specific step of the PCR. It's specific to the gene we are interested in. Then we have extension. So extension occurs via Taq polymerase, via the enzyme, which is heat stable. And what it does, so Taq is right here, this is the enzyme, is that it takes the dNTPs. Remember, it is the dNTPs are like the bricks, the blocks for building the house, and it adds the dNTPs. It looks at the, what base is here. So if there's a guanine here, it adds. If there's a GTP, it adds CTP, G to C, and in both directions. Okay, in both directions. This is extension performed by Taq, and then this cycle is repeated. So all of these, they're repeated again. Denaturation, annealing, and extension repeated 25 to 35 times. And at the end, here it shows 2 copies, but we actually have, billions of copies. We have billions of copies of specific DNA. Specific DNA. So at the beginning, right here, this DNA is huge. Okay, there are many genes, but here we ended up with specific DNA. DNA, for example, that we need to look at later on to analyze. All right, guys. So as I told you guys before, that PCR has several cycles. It goes from 25 to 35 cycles. And in each cycle, there are 3 steps: denaturation, annealing, and extension. This is one cycle. Okay, so let's say here we are amplifying a specific sequence, the eye color sequence, the hair color, or the cancer gene. So in each cycle, we have an exponential increase. We have an exponential increase of specific DNA. And after about 36, 35 cycles, we have billions of DNA. Okay, so here, cycle 1, we have 2. Cycle 2, we have 4, etc. Cycle 3, we have 8, etc. Okay, and this is a typical PCR program. So these are the steps. So let's say we do 95 degrees denaturation for 30 seconds, annealing for 30 seconds. The annealing temperature, it depends on the primer, how strong the primer is. And then we have extension to extend by the Taq polymerase. So these initial steps here, they are extra steps. Initial denaturation, 95 for 2 minutes. Okay, an extra denaturation step, and we have an extra extension step at 72 degrees Celsius. As I said, guys, PCR amplifies the target gene exponentially. So you can see here, after each cycle, how many copies we have. Okay, you can see how many copies we have. So we have an exponential increase. And here, as indicated by the graph, after about 30 cycles, we have a plateau. Okay, it plateaus out, so we have no more overall increase. So that's why people, when they do PCR, they go up to 30 cycles, because after 30 cycles, there's not much of a difference and contamination starts to occur. Are some factors that can negatively affect PCR. So we have to be aware of contamination. We have to choose an area in the lab, okay, that is clean and ready to use and that is sterilized, because we don't want to amplify other material. We don't want to amplify DNA. Sometimes you're working with a pipette, a micropipette that you use for other experiments, and then you have template DNA that is not relevant to yours. Okay, so you have to avoid contamination, clean the area, sterilize the area before you perform PCR. Also, we have to be aware that Taq polymerase, it has limits. So Taq polymerase, it has no 3 to 5 prime exonuclease activity. So it can make mistakes. It cannot go back and fix them. Okay, once it makes a mistake, it makes a mistake. It cannot be corrected. So we have to be aware of that. So it has a high, Taq polymerase has a high error rate. Let's say we got the billions of copies of PCR product, the cancer gene we are looking for. Now we need to visualize it and we need to diagnose the patient. Okay, so we need to diagnose the patient. If they have this cancer gene for leukemia, that means they have leukemia. If they don't, that means good, they are healthy. Okay, so we need to perform gel electrophoresis. So what is gel electrophoresis? Gel electrophoresis, it is a technique for the visualization and analysis of DNA. It enables us to visualize DNA using UV light in our case. The DNA fragments, you add the DNA to a gel, and I'll show you guys how this gel looks like. Then the DNA pieces or the DNA fragments, they are separated into distinct bands. They are separated into distinct bands by size. Okay, by size, by how many base pairs they have. Okay, how many pairs, how many complementary base pairs they have, okay, in a gel matrix by applying an electrical field. The direction of movement is from negative to positive. Let me explain, guys. So this is the gel. Okay, I'll explain later on how we make this gel. This gel, okay, it's an agarose gel. The agarose is the chemical. It has wells. It has holes where we can add the samples, the patient samples. So this is, let's say, patient sample 1, here patient sample 2, patient sample 3, etc. So we add the sample, okay, we add the DNA sample. Now, this gel, I'll explain in the next slide, is porous, it has holes. So the smaller DNA, the smaller pieces of DNA will go faster. The larger pieces of DNA will move slower from negative to positive. Okay, from negative to positive when you apply an electrical current. Why negative to positive? So negative because DNA is negative because of the phosphate groups. It will move toward the positive charge, the positive electrode. However, the larger ones, the larger DNA molecules, they will be slow. Okay, they will be slow. The smaller ones will move faster. So they separate by size. So how does it work? Agarose is a chemical that we use. Okay, this is the chemical that is used to make the gel. It is made from a highly purified seaweed. So agarose has been extracted from seaweed. And as I said in the last slide, smaller DNA fragments migrate faster and more easily through the porous gel than larger molecules. Okay, let me explain here. So if you guys were to look inside this gel, inside the agarose gel, you will see pores, different pores. Okay, these pores are small. So let's say these are the DNA molecules. We have different sizes of DNA molecules, larger sizes indicated by the larger ball, and then we have the smaller sizes. So they want to move from negative to positive. Negative here on top, positive on the bottom. So the smaller ones obviously will face less resistance. They're able to move faster from the negative to the positive, no problem. Okay. However, the larger ones right here, the larger ones, they will move slower because they can't fit inside the holes, inside the pores. So they will move slower. So after agarose gel electrophoresis, what you guys will see is that the smaller ones, now we have different levels. So the smaller ones move the most toward the positive charge. The larger ones will be stuck near the wells or near the holes near the positive charge. And then we have molecules that are in the middle between the smaller ones and the larger ones. So let's say we add our samples to the gel. We start the machine. When we add, and I'll show you later on, when we add our samples to inside the holes, okay, so we add them near here, the negative electrode. So this is the negative electrode. Okay, it is the cathode. It is black. This is the negative side. So we add the wells, the holes near the negative side, okay, because our DNA is negatively charged, and then the DNA will move toward, okay, the anode. The anode is the red, which is positively charged. Okay. We leave it there for a few hours until the DNA has moved down the gel. So how do we visualize the DNA? We ran the DNA on the gel, that's great, but we still have to be able to visualize it. So if you have not added a specific chemical to the gel, you'll not be able to visualize it. What is this chemical? This chemical is called ethidium bromide. So this is DNA. Let's say this is our DNA sample that has run into the gel. Okay. We add ethidium bromide, which binds to DNA, to certain parts of the DNA. So this is ethidium bromide in black. And then once you shine UV light on the ethidium bromide DNA complex, you'll be able to see the DNA. Okay, it will fluoresce and you'll be able to see the DNA. So we have to use ethidium bromide. It binds to DNA and fluoresce under UV light. Ethidium bromide can be added to the gel while you are making it or after the gel has been made. Okay, so you can add it inside the gel while you're making it or after the gel has ran. Ethidium bromide is a known carcinogen, so we have to be careful when we are using it because it's a known carcinogen, and so you have to wear gloves while using it. So you have to be very careful. So now, let's say our gel has ran. Okay, we are done. We need to visualize the DNA, so we take it into the machine, a chemi imager, and we shine UV light. And you see here, guys, we can see now the DNA bands. We can see the DNA bands, and then we can take a picture and print it out. So you can take a picture and print it out, and we can see the different DNA bands. This is patient 1, patient 2, for example, patient 3, patient 4, patient 5. Okay, the first sample here is a DNA marker. I'll talk more about it. What is the DNA marker and how to read this, this photograph. All right, guys. So let's say the PCR, let's go back a little bit, and let's say that the PCR reaction is done. We take it from the machine. Now we need to add it to the well. We need to add it to the holes inside the wells. We cannot just take the sample from each patient, for example, patient 1, 2, and 3, and just add it directly, of course, using a micropipette inside the wells. We have to prepare the samples. We have to add a loading dye. Okay, so we mix the samples of DNA, of each DNA sample, with a 6 times loading dye. So this is an example of a loading dye, as you guys can see here. It's bluish in color. It allows the sample to do 2 things: to be seen, to be tracked when loading onto the gel, and to increase the density of the samples, causing them to sink into the gel wells. So what that means is that it has two components. It has the 6 times loading buffer has two components: a bromophenol blue dye for tracking, okay, for tracking the DNA. And what that means is that it allows us to see, not to visualize the DNA, that will be for ethidium bromide, but to track the DNA, so that when it is run on the from the negative to the positive side, we can track to see where the DNA has went. Okay, because we don't want our DNA to fall down from the gel. Okay, this is for tracking the more bromophenol blue component. And there's another chemical in the loading buffer which increases the density of the samples. Okay, it allows the samples to sink in. So for example here, if you are loading the sample patient samples inside the wells, if you do not add glycerol, the sample will float in the buffer here, covering the gel. So glycerol increases the density, the weight of your sample, and makes it sink in, go inside the wells and sink in. Okay, otherwise you will lose your sample. So the glycerol part is for allowing the sample to become heavy so that it falls down inside the well. This is how we make the gel, guys. These 2 slides, or the next 3 slides, it shows you how to make the gel. It's not really important, but just here, notice that they use ethidium bromide. They add it while they're making the gel, and they use a special tray. Okay, they use a special tray. You don't need to know this, but just the main things here is that, so notice here how they're adding their samples. They added the loading dye. So each of the patient samples, so this is patient 1, patient 2, patient 3, let's say, they're adding the samples into the wells. Okay, but before they added their samples, they added the loading dye. Notice it is blue so that it can sink in and they can track it. Of course, they already added the ethidium bromide inside the gel. And then they turn on the power and it will run from the cathode to the anode, negative to positive. Okay, and notice here, one of the samples is a DNA marker. I'll show you guys what a DNA marker is. So you always have to have a hole containing a DNA marker, or a DNA ladder. Okay, DNA ladder, that's another word for it. All right, guys. So the main thing from this slide is that, let's say after 2 hours or 3 hours, the DNA has moved far enough. So you will notice the DNA, which is negatively charged, has moved to the positive side, the positive electrode, and it has separated based on charge. Based on size. Based on size. So you see here, the shorter molecules, let's say 3 base pairs, 4 base pairs, have moved faster toward the positive side. The larger molecules, let's say 1,000 base pairs, 2,000 base pairs, are stuck at the beginning, near the negative side. Okay. And now, of course, we have to visualize it. So we've added ethidium bromide to it. Okay, and then we have to shine UV light so that we can visualize it under the machine. Okay, so in this slide, the important thing here is that we're using UV light. Remember, we've added ethidium bromide, so ethidium bromide is stuck to the DNA. So if we shine UV light, we will be able to see the DNA bands. So here we have shown UV light on our gel. Okay, and then we took a picture, so you can see here the DNA samples. So this is, let's say, patient 1, patient 2, patient 4 or 5. We have different size bands. How do we know the different sizes? By having the DNA marker. So this is the DNA marker, and I will explain in the next slide, I believe, what a marker is. So this is the marker, or a DNA ladder. This is an example of what you might see after you run the gel. After a few hours, the gel has run far enough, you'll be able to see this. Of course, with the patient samples, you guys can see here different patient samples, you have to add a DNA marker. And a DNA marker, or a ladder, is composed of size standards. So these are samples that we buy from outside, samples of different sizes. We use them for comparison, okay? So here, this is the DNA marker. The smallest one is 100 base pair. Then you have 200 base pair. This is the unit. BP stands for base pair. This is 2000. We use it as a marker so we know the size of the band. So without the marker, we'll not be able to know the size of the band. So we know now that this one here is about 200 base pairs. This one, it's about 800 base pairs, etc. By knowing the sizes, we'll be able to find the correct band. Find the correct band and analyze it further. Okay. So let us conclude with this. Now, remember, our goal for this experiment is to find which patient or which patients have the CML type of leukemia. Let's say we got 8 blood samples from the hospital. We don't know which patient has leukemia. We isolated the DNA in experiment number 5. We have pure DNA. Now, in experiment number 6, what we did is we did PCR to amplify that specific piece of DNA. So we're investigating if the patient have CML type of leukemia. It's a cancer of the white blood cells. And let us assume that the size of this leukemia gene, the BCR-ABL gene, is about 600 base pair. Okay, let's just assume that. And so we're going to look for this size. If we can find this size, we can diagnose the patient. Okay, we can diagnose the patient with leukemia. Okay, of course, guys, this is oversimplification. It's more complicated than that, but let's assume for our purpose. Okay, so we look for this size when we run it on a gel electrophoresis. So this is how the result might look like. Okay, so here you have a DNA ladder. We have actually 2 DNA ladders, 2 DNA markers for comparison with different sizes. The sizes are base pairs, and we have 8 patients. So can you guys tell me, please, which patients they have the CML type of leukemia? Which ones they have the CML type of leukemia, assuming that this is the size, this is a 600 base pair band. Okay, yes, so you can see here, guys, patient number 1 is positive, patient number 4 is positive for leukemia, patient number 7 and patient number 6, I would think so, but I might have to do it again. But for sure, patient number 1, patient number 4, and patient number 7. Okay. Of course, patient number 2 is negative, patient 3 is negative, 5 is negative, and 8 is negative, because if there was a cancer gene, it would have been amplified in PCR, because we added the primers specific to the cancer gene, but it was not amplified, meaning that these patients did not have leukemia. And this is, guys, this is how we can diagnose patients using PCR. One possible use. Of course, this is, I'm mentioning again, this is oversimplification, but it gives you an idea. Thank you guys for listening to the presentations. Please go back and answer question sheet for experiment number 5 and number 6. It should help you or check your understanding of of your understanding of the lessons. And please email me if you have any questions. Thank you.